So welcome back everyone to the second part of today. We are going to be starting this session talking about Coorong water quality, aquatic plants, modelling and integration. We have three presenters for this theme area. And again, a reminder, please hold your questions to the end of the three presentations. We have some dedicated time for the Q&A. Of course, to the people online, please feel free to enter your questions in at any time and we will get to those during the Q&A session. So our first presentation today, I'd like to welcome Luke Mosley, who's going to talk with us about nutrient dynamics. Thanks, Luke.
Thanks and good morning everybody. Happy World Wetlands Day. It's a pleasure to talk to you today about nutrients in the Coorong. So I'd like to start by saying nutrients are critical for all aquatic systems and wetlands in particular. So it's not like nutrients in and of itself are a bad thing. The problem is really when we get too many nutrients in the system, and that's when you can lead to problems, because nutrients really provide that fuel or food for plant growth. So you can think about it like a fertiliser. When you put too many nutrients in the system, there's too much algal and plant growth, and Michelle will talk more about that. And some of the symptoms, I guess now in the Coorong is a very nutrient and organic matter enriched system, an algal enriched system. So you can see that photo on the left there with a lot of algae floating in the water and a really sort of soupy coloured water, indicative of a lot of algae and organic matter in the system. And like we've talked a lot about today, everything's connected. It's the same with nutrients. So nutrients that get taken up out of the water and go into algae and phytoplankton also can end up back in the sediment where they're broken down by bacteria. And one of the symptoms of high nutrients in the sediment is production of these really thick black organic rich oozes, which again we see in the Coorong today. Today I'm going to talk to you about part of a project I'm leading called the Nutrient Dynamics Project as part of the Trials and Investigations component of Project Coorong, where really we're trying to gain improved knowledge on several aspects relating to nutrients. Firstly, where nutrients are coming from and where they go in the system. How nutrients are cycling, so what happens when nutrients come into the system? How does it flux between back with the sediment and water and out to the aquatic plants? And we're trying to use this knowledge to inform nutrient removal options and ultimately to achieve what we're all trying to achieve, which is shifting the Coorong to a healthier state. But we're really working at that fundamental water quality chemical level of the system. What we know today, currently there's, I guess, been a long-term monitoring program which has sort of got started in late 90s and has had a few gaps along the way, but certainly has provided useful data. I'll show you some of that data in a minute, where there's samples been taken along the Coorong at multiple sites and we've been able to look at nutrient concentrations over time. This just shows you a graph of, firstly, it's probably a little bit hard to read, that's salinity or electrical conductivity there, that's total nitrogen and that's total phosphorus from 1998 to essentially 2020. And this is Long Point in the North Lagoon, the blue site, and the black dots are North Jacks Point in the South Lagoon. So you can see the South Lagoon always has higher salinity, obviously, but also higher nutrients in the system. And the other thing I'd like to point out to you, we've talked about the millennium drought today. This is the millennium drought period here. And you can see a lot of the total nutrients really going up in the system. So nutrients were really being retained within the system, in the water, but also in the sediment. And when another way to perhaps look at that is to plot the relationships with salinity. So taking all the data from all the sites and plotting nitrogen, total nitrogen on the left and total phosphorus on the right versus salinity. Seawater's around just over 50 millisiemens per centimetre EC. So you can see once you increase over seawater salinities that the nutrients really start to increase and really ramp up to some extremely high levels. So that extreme hypersalinity is linked to high nutrient concentrations, and that's really symptomatic when you get a lack of flushing of the system, when you don't get enough water coming into the system to flush the nutrients out. They can accumulate in the system and you can build up those high concentrations in the water and also, as I'll talk to you now, in the sediment. So essentially when you get high nutrient concentrations in the water, some of that, I guess, algal material and all the material that's in the water can settle back to the sediment. So having high nutrient concentrations in the water will also tend to lead to high nutrient concentrations in the sediment. And that will drive that poor health that we're seeing in the South Lagoon in particular today, where we've got these really black oozes in a lot of places that are really high in nutrients and organic matter. And the lack of benthic invertebrates, I think Sabine will talk to you a bit more about this, but we believe that's also having an effect, because where we see benthic invertebrates present that create burrows, they bring oxygen into the sediment. You can see some of these burrows coming down here. That helps to break down that black ooze. And similarly with rupia, the roots can provide oxygen to the sediment and help to improve the quality of the sediment. Some of the research that we've been doing is linking, I guess, where nutrients are going, and we've seen quite consistent signatures with what's in the water and what's in the sediment, so that isotope signatures of that material. So we're really trying to trace where nutrients are moving, and we're also looking at how the sediment is then providing nutrients back to the water column, and also groundwater investigations where we're looking at what's coming in from the groundwater on the margins of the Coorong. Some of the, I guess, early research findings is also, as well as those high nutrient concentrations, we're actually seeing quite unhealthy nutrient cycling in the system. We're seeing very high nutrient concentrations in the sediment, and that is also fluxing back to the water column. So we've been doing a lot of experiments where we take sediment cores, we take them and we incubate them in these incubators, and we measure what's coming out to the water column and also what nutrient concentrations are in the sediment. This shows you, this is depth in the zero is the sediment surface, this is the water column, and this is down about 13 centimetres in the sediment, and this is ammonium concentration. So you can see very high concentrations here, and that gradient is indicating a flux essentially to the surface water. So that ammonium is fluxing out to the surface water and providing that fuel and those nutrients for algae to grow back in the water column. So it's really a vicious cycle. You start to accumulate nutrients, they start to flux back, that fuels more algal productivity, that algae dies eventually, goes back to the sediment, and you start to end up in these unhealthy cycles. And some of that unhealthy, really hypersaline sulfide-rich sediment is also not enabling nutrients to be lost through the normal processes, a particular process called denitrification, where nitrogen comes out of the system as a gas. So that's quite important, where you can actually lose nitrogen out of the system, and that's not happening in the South Lagoon at present. And there's some good research being done by some researchers on our project on this. So the whole story is really nutrients are being retained and recycled in the system and leading to an unhealthy state. So we're trying to, I guess, unpack all these cycles and really understand management, how to manage the system and what needs to be done to improve the system. And one of the things I guess we've talked and fed into some of the investigations that the department are doing in relation to infrastructure and other feasibility investigations is the need to improve system flushing. So if you don't move nutrients out of the system, they will accumulate and they will cause those problems. So we have to get better flushing of the system. And we're also improving the biogeochemical models to enable better predictions. So all those nutrient processes that we've described are helping to improve all the models that the government can run scenarios, and then we can sort of test different options for the Coorong and hopefully reach a agreed set of way forward for management options. And I'd just like to thank, there's many people involved in this nutrient project, several who are here today, and thank the team and community members and also the department and Goyder Institute for their support. Thank you.
Thanks, Luke. Sorry, I was caught out because I had you had at least a few more minutes, so well done for keeping to time. All right, I'm now going to introduce Michelle Waycott, who's going to talk to us about Coorong Aquatic Plants. Thanks, Michelle.
Thank you. Thanks for joining us here today, World Wetlands Day, but also to come together to talk about a place that we all believe needs some protecting and caring for, and that we're all happy to work together to do that. I'm going to talk about aquatic plants, and I just wanted to start by emphasising to you all, when I talk about aquatic plants, I talk about aquatic plants on a whole range of different scales. The macrophytes that we've heard lots of people talk about, particularly Rupia, and I've got this weird thing up there called Althenia. Some of you may have heard of something called Lepelina. Lepelina is now Althenia. So in the best tradition of taxonomists, go taxonomists, it's got a new name because it's actually very closely related to something from Europe. But I'm also talking about the primary producers of the system, the microphytes such as the phytoplankton, the benthic microalgae, the micro plants that grow on other plants or other structures, cyanobacteria, diatoms, dinoflagellates, all these organisms are part of the aquatic plant group. And we're just sneaking in a little bit about the other microbes as well that may or may not be photosynthetic. So it's a very broad group of organisms, and what we're trying to do is understand the dynamics and the changes of these organisms in the system to help us improve the health of the system. When I talk about the health of the system, we've got some various conceptual diagrams that lets us sort of visualise that. I'm not going to spend very long on this, we've talked about this before, but the current state of the system, which I've summarised at the top here, I've very carefully drawn the system to be heavy in the water column green, sort of nasty green colours, not happy green colours, and these black blobs on the sediment, which are these black oozes that Luke talks about, and that's the product of this high organic load that forms on the sediments and is starting to form down through the sediments over time. And the North Lagoon's been drawn here all happy, pretty light blue colours. It's not quite that happy and pretty, but in comparison, the water column and the sediment processes are quite different, and I think that this captures it. But variability in this system is highly expected, and over time, a historic version of the system, which has more birds and more of the macrophyte aquatic plants, through a whole range of different stages, what we're dealing with as well. So depending on the year you're out there and depending on the leading years and the following years, the system can change. And many of the talks that you're going to hear today are going to be people who are documenting and discussing that. And I go back to something that Tim and Candice spoke about from the Naranjiri perspective, that is, everything's connected. And when you work on organisms as a part of the whole system, you very quickly need to recognise that everything is connected. We have a conceptual framework that we're testing, and Matt Gibbs is about to talk to the hydrodynamic side of some of the modelling work that we're doing in this program. But all of those things come together, and from our perspective, we have the health of the aquatic plants in the middle there as our desired goal is to improve that. We have lots of data sets that are coming together to give rise to our current view of that. This plot here recognises there's a number of different ways that the key species that we're trying to return to the system, the Rupia, has 3 different pathways it can survive through the year. It can go via seeds, it can have its turions form, or it can even hang in there just with bits of vegetative growth in the water that doesn't go out too deep. But these plots here show a whole range of conditions over which they can survive. In this case, the salinity ranges, and you can see some of the tolerance ranges are quite extreme. So the program of work that I'm leading for the Coorong aquatic plant component has 7 elements to it. I'll start at the back end, and that is we're working closely with Luke's team on nutrients, and we partner on a number of those projects. We're looking at a quite active project I'll talk in a minute about on the microbiota communities. And then we've got phenology, some experimental testing to feed the modelling that you'll hear about, and then a restoration strategy as well as baseline understanding of what's in the community through the 4 seasons of the year, including data that'll feed into Chief Feng's food web modelling, as well as some of the other projects. So a little bit of preliminary data. Our spring last year, some of you may have already heard some talk in the media about this. The rupia is now returned to its pre-drought extent. So the red dots on this figure are a big tick for, yes, there's rupia present. I know the colours are back and front, but it's all right. The blue is where there's no rupia, and that's basically where there's hard substrates, or where the substrate's quite friable. So rupia is very widespread. But what condition is it in? And so some of the measures we're taking side by side, this kind of survey data, are to observe things like how the temperature is fluctuating actually on these meadows as the water comes in and out over the year. So we've got these little temperature loggers actually in the areas where the meadows are growing, which can augment the long-term monitoring stations. And what I think is pretty cool is that when you look at this monitoring data, you can see the times of the year when the water level drops out and where the rupia beds are growing, the sediment becomes exposed and dries off. So we can get within a couple of days where these orange arrows are, where the habitat's drying out. And this is 3 different locations across the system. We also can see the return of the water. And what's really striking is the water comes in earlier down at Salt Creek end. And I'm assuming, I haven't actually done the exercise yet, but I'm assuming that's because of flows coming in through Salt Creek, helping the water levels start to come up in that shallow water quite quickly. This area is quite close to that. So this kind of data, in addition to our observations, starts to let us unpack what's going on in the actual places where the plants are growing. You've heard a lot of talk about algae and the importance of this shift in the community composition. One of the problems with the algae forming is that it's not that it wasn't ever a part of the system, because these species of algae are very common amongst estuaries around the world, and particularly along our temperate coast. There's a couple of endemic species, but then there's the one that makes this look really gross. It used to be called Enteromorpha intestinalis, its old name. You can probably see why. It's got a different name now. It's now called Ulva-paradoxa, but it's the same species. And it's what we call a nuisance filamentous algae. It grows when there's lots of nutrients freely available. It grows incredibly rapidly during the time of year where the temperatures go up and where there's excess nutrients. And so Luke was talking about the sediments having unhealthy organic loads. Some of it comes from this, and on a macro scale, this is one of Jeff Galash's images where we did some clearing of algae. We cleared about a hectare and it got covered up pretty quickly. But you can see that these areas are very extensive. And in fact, summer before last, there was more than 10 square kilometres of these filamentous algal mats in that central section of the Coorong, roughly calculating to more than 100 tonnes of dry weight of algae, which is a massive amount of biomass, which is driving some of this nutrient values. So the next stage of our work is to look at the micro communities that are in there. And Sophie Leterme from Flinders Uni is working with us, and we're sampling not only the water column or the sediments, but we're also sampling the actual rupia plants or the macrophytes, as well as these algal mats. And we're trying to see what actually is the microbial or the micro community there and how it changes again over the seasons. We've also started looking at the question of these organic layers that form late in the season. So if you go out there now, there's not many living plants like this. It's mostly just kind of a black or slimy, sludgy layer over the sediment where it hasn't been washed away already, although I don't know how much is going to be left after today, and work out what's in that community. Is it starting to fill up with blue-green algae, or is it starting to fill up with bacteria or fungi? These are questions that we can't answer at this time. Coming back to Luke's question about the Ruppia root zone, and this is one of his photographs he showed you earlier. This interface between what the plant's doing, where it photosynthesises, creates oxygen, pumps it into the sediment, and you probably can't see it very well, but these areas where you can see that oxygenation of the sediments happening, reducing the dark sort of grey or black sediments, making them more oxygenated, is also an area where a lot of important biochemical processes that feed both the sediment, allowing other organisms to live there, but also feed the water column above it, enabling these plants and the other organisms that should be living in there, invertebrates and fish, to grow. These plants, I'll just flick through that. The critical importance of this root zone needs to be really well recognised, and so to do that we're looking at what the taxonomy of the communities in there are. So what are the, in this bit here, there's a plant that's been dug up, these are the roots which would normally be under the ground, and looking at what communities are growing on it. Are they important? How are they? And when do they turn over? So what's the current state of the aquatic plants in the southern Coorong? This is Jeff's hand showing the nice sludgy stuff. And there are lots of places where if you were to go out in late spring, early summer, instead of a nice healthy Ruppia bed that's setting lots of seed, what you get is this kind of mess. And this is symptomatic of the excess nutrients in the system and the inability of the macrophytes to absorb those nutrients, or the system to flush to get it out. So what this is our current state, what our desired state might look like is an increase in flushing here, reducing those water column loads and increasing the biomass of these plants, which help bring in the community. I've finished in time. Well done. I just want to thank my team. They're great and very committed and put up with lots of not very pleasant field work at times. So thanks for your attention.
Thanks very much, Michelle. All right, I'd now like... Is that right? Or have you got a few extras? There we go. Here we go. I'd now like to introduce Matt Gibbs, who's going to talk to us about hydrodynamic modelling and integration. Sounds exciting.
It is exciting. Yes, thank you. I'll talk about the modelling we're doing as part of this program, and also touch on the water quality monitoring that the department's got underway, just to give you a feel for what's happening out there now. And the main thing I want to get across in terms of modelling is almost everything you're going to see today around the science and what Luke and Michelle have just presented, as well as after me. We've got a home for it in the model where we're eagerly awaiting the data and the analysis to feed it in somewhere. We've got three sort of broad categories that it fits into. The first one is the model itself, how we set it up. We've got to put in thousands and thousands of lines of code with equations and numbers. We need to actually specify all of that. So a good example is the life cycle that Michelle put up. There's salinity ranges, there's times of the year, there's water levels, there's temperature thresholds, there's light availability. So we can put all that into the model to interpret the results and say how suitable will it be for Ruppia. And that's just one example of many. We showed the sediment processes, we're trying to represent them. Everything is in there, the information that's been collected, the data, really helps us set that up. The next one is the boundary conditions, so what we force the model with, the outside conditions that we drive it with. So good examples are the meteorology, so that's the Parker Point weather station there on top of the crow's nest. This is Luke's sediment profile, so we need to tell the model what is the sediment everywhere across the Coorong. So we need to get all of that in, driving the model so we can replicate what happened inside. So that's sort of the second category, is these forcing conditions that we drive the model with. And then the last one is how do we assess it and make sure it actually works. So we've been working on these models for a while now, at least 10 years, probably longer. So we're looking at water level here, and it's doing a reasonable job of matching water level. But there's a lot of other parameters. The biogeochemistry and the water quality is a fairly new one. We know needs some work. So as we get those measurements in, we can check that the model's working. And that's really the key to all of this. If we can set it up like that, it demonstrates that we actually understand what's happening. If we can take our knowledge of the system and put it into the model, it's a good indicator that testing our understanding of the system. And then if we can get that, it helps us answering all the management questions. So from the short term, how do we manage the barrage flows over the next few months, to the longer term, the big infrastructure questions. So that's really what's happening in terms of the sort of the more researchy side, the science side of the modelling. Really spending a fair bit of time on these models, getting all the data and knowledge into them, and checking that they work, so we can use them for the decision-making parts of the project and the department more broadly. So that's really the main point I wanted to make, but I couldn't come up here and talk to you without showing some sort of results. So this one, it's a little bit confusing, but the yellow is water that starts in the South Lagoon, and there's some little green floaty balls in there if you can see them. And we'll model nearly 3 years forward in time to see where that water that started in the South Lagoon goes. So hopefully that's going to work. So yellow is South Lagoon water, blue is not South Lagoon water, and sort of in between is a mix. And what we see in this run, which is 2013 to 2016, pretty low flow period. Not much, there's flow coming over the barrages, but not sort of the 2016 floods. You can see there's only sort of this time in spring, we're coming off the winter high water levels with low pressures and storm surges. It's only when that drops that we actually get some of that yellow getting all the way out the mouth. For the rest of the year, it's just sort of sloshing back and forth, sort of in the South Lagoon, into the North Lagoon and back again. So I'll run it through again, but it's really just these periods now where we sneak a little bit out, and otherwise it's just sort of sloshing around back and forth. So over this, almost 3 years, we've got a third of that initial sort of lump of water actually out of the system. So we've heard a few times today about the accumulation of the system and how it's a problem. But this really brought it home for me of just how much or how little really we managed to get out under these low flow periods. Making that point a different way, sort of moving into the monitoring side of things. This is all of the salinity data from the continuous stations at least in the South Lagoon. So we have the Sandspit Point site, it's closed now, but that's sort of south of South Salt Creek, and then the two current ones at Woodswell and Snipe Island. So you can see the Millennium Drought there pretty clearly. Over here, salinity is going up. These black dots down the bottom, if you can see them, are periods of high barrage flow, 500 gig in a month. So that's almost beyond our ability for environmental water and regulated conditions. That's not flood, but it's high flows. But that's really the periods when we see the salinities come down again. And then when that stops, salinities start to increase. Here's 2016 high flows, salinities come back down, and since then it's been creeping up again. So it is those high flow periods naturally that manage to deflush the system. And as we've seen them become less frequent, we see this accumulation problem that we keep talking about. So it's pretty clear from the modelling and pretty clear in the data we've got. That's sort of how the system's behaving. But for the rest of the time, I'll just talk about the monitoring that's happening, that's going on now. So the guys out in the field, if you see Pat and Josh, they've done an amazing job trying to get some water quality, continuous water quality monitoring happening. These are pretty finicky little things. this is how they come out of the water, all covered, all very dirty, not really working very well. So they've spent probably the last year going through different sensors, trying to get them to give two of the same sensors next to each other to get the same measurements. Very difficult sort of system to work in. But over that work and testing and trying different instruments, different manufacturers, we think we've got a pretty good system now. So basically we've got a backup set. We'll have these in the water, and then when they come out to maintain them about every month or so, they'll have a second set that's been taken back to the lab in Adelaide and cleaned under the right conditions. They can swap them out. So we're getting nice, good quality, continuous data for us modelers, and it's not. They can be a bit more confident in what they're doing. So these will be at 4 stations across all of the Coorong, and it's giving us measurements of dissolved oxygen, turbidity, pH, and a measure of dissolved organic matter. Tells us a little bit about the algae and where the water came from. On top of the water level, the temperature, and the salinity that we've already had. So it's been quite a good effort. Lots of testing and trying from the monitoring guys, and we're pretty optimistic that we're going to get some good data now. They're just going out now-ish, I think. But we can't use those sensors for all the water quality parameters. So this is the stuff where we grab a bottle of water and send it back to the lab to be analysed. So there we get a heap more parameters that need that more detailed analysis. And while they're out doing it, there's Pat there with the sensor, getting a depth profile of dissolved oxygen, salinity, and light. That's a really useful one. So also checking how things are changing with depth, if we're seeing any stratification or not. We tend not to see it too much in the historical data when they have been doing it. But how the light penetrates is another very useful one. Yep, sure. I mentioned meteorology. This is the weather station at Parker Point again. So obviously the wind speed and the temperature and the solar radiation as well are really useful parameters. So we've got one of these at Parker Point. There's one going in at Long Point. And there's another one just inland at Policeman Point, just out of the South Lagoon. So we're going from having just one station up near the barrages historically to having 3 more in the Coorong now, which again really helps us drive the models, understand what the changes are. So we're really beefing up what we know about the system. And just finally, the last little cool bit of kit we've got out there, just near Twitchery at Pelican Point, is one of these velocity sensors. So it's in there on the probe permanently and measuring which way the water's going and how fast it's going past. So it's, again, everything's useful for the modelling. We're data hungry, but it also helps us with the real-time barrage operations to know which way the water is going. Is the tide still coming in? Has it turned around yet? So this sensor's giving us that. And this bottom one here is the model looking pretty good, I think. So they've got a little body board, if you can, red thing in the photo over there with a velocity sensor on it. With that one, they can get the flow. So over spring of 2019, they were out fairly regularly, measuring the flow across Parker Point, and we've got the model to represent that pretty well. So this is sort of the process. We're collecting a lot more data. We're challenging the model with it to make sure it can still work, represent those conditions. And we're doing this, more and more detailed, more and more parameters over the next year or so. So here's just a map of all the bits and pieces that are going to be out there now. A bit hard to see, but the red dots are the grab samples. The blue are the MET stations. Green is where we're going to have the water quality probes, the sort of fouled up ones we saw in the photo. And the black is the existing water level, salinity, temperature sites. So that's it really for me. It's really bringing home this point of all this science, it makes its way into the models. We're really hungry for it. We can make use of all the data available, both past and present. Improves our understanding of the system, or challenges our understanding of the system. Can we actually replicate it in the computer, what's happening out on the ground? And then that really, the main point is to test what if questions. What if we do this? What if we do that? If we've got a well-validated, trusting model, we can do those sort of things with more confidence. And then just to bring your attention, I guess, that the monitoring is happening. A lot of that will be available, in real time on Water Connect, and it's a new system that's coming online soon. So yeah, a lot more stations, a lot more parameters available now. Thank you.
Thanks very much, Matt. And again, thank you to all our presenters who spoke with us today about theme 2 relating to the water quality aquatic plants modelling and integration in the Coorong.